in today's session, today's class, I will discuss about plant authentication. As you know, plant authentication is one of the vital parameter for any kind of research work, as well as the moment you are carrying your information and something related to the pharmacological profile of any herb. So as far as the authentication of the plant is concerned, first of all, you have to know the morphological aspect of the plant that you have already gone through in previous semesters. I guess in fourth semester of pharmacology and phytochemistry 1, there was morphological evaluation where we have studied about the collection of the plant as well as the morphological aspect of the plant. It means if you are supposed to collect root or leaf, or in case of the collection of the entire plant, how do you go with the drying process? Before that, even when the moment you are collecting the plant from different areas also on the basis of their distribution, geographical distribution. So few points are there, and those points are related to the collection of the plant. Means if you are going to the mainstream authentication, the first point you have to focus on, that is nothing but collection of the plant. From which area you have to collect the plant, and it totally depends on the previous information that you are gathering regarding the plants. And this is very much important for the research work, because if you are collecting same plant from the different areas, it may vary the same chemical constituent but in different percentages. As you all know, it depends on the cultivation factors, it depends on the soil factors. Of course, soil factor, climatic changes, and kind of parameter that is associated with the weather of different areas, that also depends related to the cultivation of the plant. So the moment you are collecting the plant, you have to identify the plant very firstly morphologically, whether you are going to collect the particular part of the plant or the entire plant. In case of herbs, specifically if I am considering herbs, because we are going to the subject of herbal drug technology, so I will discuss about the herbs. In such cases, you have to, because herbs are not bigger in size than you want. So it will be collected in a smaller form, and then it will be processed, means you have to remove the dirt and all, then going for the drying process. The moment you are starting the drying process, it's related to the preferable portion for drying process is concerned as the shade drying. Okay, in normal shade drying process. So after the drying process, you have to go for the means planting all those products in a herbarium sheet. But make sure that before collection or in time of collection, it will be better for you if you collect your plant material in its full blossom area. Means if it is a flowering plant, then it should have, if it is possible, then it should have its flower, its fruits, along with the seeds, and its full blossom area. Means it is applicable more specifically for the seasonal because there are several herbs are there, or plants are there, which grows most abundantly or in its foliage in certain season. It depends, because if you consider the spring season, so in spring season in Ganges area or eastern part of India, if you consider the plant of neem, the new leaves of neem we used to observing they are growing, new leaves of neem. So I mean to say that if you are targeting the particular season, you have to collect the plant in particular season, wherever seasonal plants are applicable. Whenever all season plants are okay enough, for example tulsi, okay, no problem at all in such cases. So in those cases, it totally depends on how do you relate with the product, means the sample you are collecting, and in which season you are collecting. Second, in which month, which date you are collecting, and in time of collection, what is the color, what is the present appearance. That's why I started my lecture today along with the morphological aspect of the plant. It means the moment you are collecting the plant material or herbs, what is the color of the leaf or stain or the entire plant or different alien parts that are those are exposed to you in terms of collection. What is appear to you in which color? Whether it's in light green color, pale yellow color, yellow greenish color, or any other color, deep green color. In few cases, you will find that upper surface of the leaf that is having dark green color. Lower portion of the leaf it contains light green color. So the moment you are writing the different parameters or different observations in the harmonium sheet, you have to mention all those. In time of collection, the moment you have collected the plant in time of collection, the upper portion of the leaf was supposed to be like dark green color. The lower portion of the leaf it was something like pale green color. So that is how it will be helpful for the taxonomist to identify the supplied sample for proper authentication. Now you have to know what is the importance of the authentication as far as the CO and PO is concerned. Along with those topics, the main outcome of such topics lies in the research specifically, because you know that you are dealing with certain single plant that is crucial, basal, and whatever it is, or aloe vera. But whenever you are aiming towards the market, towards your customer, those are buying the herbal products or Ayurvedic products from your company, they must go through the protocol of safety very first, because WHO they are mentioned in their guidelines that whatever drug or nutraceuticals you are using, that should be safe and pure enough for the entire mankind. And on the basis of that, regulations or Schedule T protocols, whatever we are maintaining, by maintaining the law governed by WHO, they are, or even in India, Ayush, the Department of Ayush, they are undertaking all those. So they all are under one umbrella that product should be authenticated enough so that it will be pure. So whenever somebody is authenticating your product from government background or from regulatory background, the customer or the consumer will be safe enough, feel safe. Means whatever product I am using, it may be manufactured from Vaidyanath or Zandu or Dabur India, doesn't matter. Whatever company you are using doesn't matter. In the middle, what matters, your product is safe enough to use. That is important. Because in case of Ayurvedic medicines or herbal medicines, you may appear to the different specific practitioner, and by showing the prescription, you are buying this product. But in case of nutraceuticals, either your nutritionist is prescribing all those products, or your dietitian, or even sometimes we are just purchasing the product by ourselves. Means by our social knowledge or environmental aspects, whatever we have seen or whatever we have gathered knowledge or experienced about the product previously, someone else or from your family background or anybody else. So it's a combination of whole. So the moment you are purchasing a product by yourself, you are also just observing the product like, oh, this is the product and this is the manufacturing company, this is the marketing company, and this product contains this kind of contents. So if it is supposed to be plant product or any herb, it means it is previously authenticated. Then only they are capable enough to sell the product in their showroom or in their shop, medicine shop. Okay. So it means the moment you are authenticated, you are just authenticated the product, it is applicable from manufacturing as well as for research also. So that is the importance of authentication before going to the mainstream production of the product. And another thing is that in case of certain novel drugs, for example, X is a novel drug. Often you have just kind of newer molecule, you have isolated the molecule from a herb or from a plant. So the entire world want to know basically what is the molecule is all about. Firstly, what is for it is functioning. For example, if it is very much potent action against a disease like diabetes or cancer, which is considered the burning disease, all over the world right now. Specifically in India, that you know, this nation is known as the diabetic hub of the world. So in such cases, people must rush before the product to purchase this product. But you have to authenticate the product first. Whether it has been authenticated, the plant has been authenticated, whatever they are claiming, the research company or the manufacturing company, whatever they are claiming, is it right? Is it safe enough to use? If it is there, then it is there. Even though, if you are publishing a paper, a scientific research paper, so you are claiming that ethanol extract of such and such plant is having potent antidiabetic activity on albuminous with fats or something like that. This is the rating of the paper. And this paper has been accepted by Elsevier or Springer, whatever. The top, even for the Nature, means topmost publishing house for the publications. But they want to know whatever data you are producing in front of us. Is your plant from where you have got the extracts of isolated carbon? Is it authenticated properly? If it is not, then it will be rejected. Similarly, same for if you are going through the animal study, so you have maintained the ICH guidelines, OECD guidelines. Is it applicable? Means it is all about the standard protocols individually. For animal study, these are the guidelines. For plant authentication, these are the animal guidelines. But whenever they come together, it will create a good paper, good research paper, good informative one that can be helpful for the mankind in terms of when it is available as manufacturing product. But before that, there is a long journey, and you have to clarify each and every section. Plant authentication done, it is okay. It has been done from a specific field, and it is acceptable enough, well acceptable, no doubt about it. Animal study, it is done. It is safe enough because I have maintained these and these guidelines. So when that will be done, your molecule will be authenticated. You have probably gone through the term structured elucidation of a newer molecule. In case of structured elucidation, such kind of authentication is needed also. Structured elucidation is something which is related with the molecular aspect. Means the authentication of the molecule, which kind of functional group it is, what it basically possesses, and kind of activity it is having, followed by the other parameters, other analytical parameter, or the exact chemistry of the product, how it will react, what will be the molecular configuration. That's why I use the term molecular term. That's why I used previously. So when the molecule is established, it is not all. Before establishing of the particular molecule, you have to observe the journey. Each and every protocol, if it has been done properly by maintaining the authentication and all, then it is okay. Otherwise, it will be done. So, up to that, if you having any question, you can ask. Do you have any question related to that question?